About time to start. So, welcome everyone to the session. In this session, we'll talk about visualization and network analysis. So, in previous sessions, we conducted summarization of the data. We conducted various analysis. We were comparing samples. We were analyzing scatter plots. We were analyzing correlations. We used regression analysis to confirm if there is association between two samples, for example. But later we used algorithms like DSEC and, for example, factor analysis. We combine them with the regression to see uh, what genes could potentially be associated with certain factors. Do certain factors, like for example, mouse model or subtype of breast cancer, like triple negative or ER plus, do have an influence on gene expression. So we conducted differential expression and narrowed down a number of genes that were differentially expressed between those samples, subtypes, or between those factors. There is another way of improving this analysis. So we're not going to be just finding different expressions across the samples or subtypes of some disease. There is also a way to use a segment of data science that is called machine learning. So this session will be also a brief introduction to some basic machine learning for you. What is machine learning? Machine learning combines statistics and computation to improve data analysis. So we have algorithms that have learning aspect. They learn from the data and find most optimized model and then give us much better results than standard statistics. Using such algorithms, we can create a network of association between, for example, genes. So last time we found out that some genes are differently expressed between the samples. Now we will use machine learning to create pathways and networks of interactions between those genes. So this will be an improvement to the analysis we did before. We'll also be conducting a lot of machine learning in our next sessions, and for now, we'll stop here and I will introduce you to some other aspects of today's session. First, let's do a brief recapitulation of uh, what we did before. Right here, you can see something about genes and isoforms, expressions. As I said, we run NGS pipelines before, so we use algorithms like PCR clean, Trimomatic, HiSet, cufflinks, cuff merge, bow tie. We created an expression table, and we will be using these a lot in most of our analysis. There are other pipelines that we will use as well, but generally this is the initial point, right? When dealing with transcriptomic data, this is pretty much the initial point. After we get the expression table, then we have data sets like the ones we have, like the ones we're analyzing, and then we can use, for example, factor regression analysis, we can normalize them, or we can use other machine learning algorithms like principal component analysis. We'll be also speaking about it in our next sessions. So I'll just recapitulate for you, not to forget those initial steps. We'll be doing this a lot of times. It's very important. So we're just repeating this briefly, right? So I'll just get back here. We were going to start, right, when you were running the demo pipeline. And then there was that Trimomatic algorithm that removes the adapters. It removes those sequences that were added from technical perspective to mark beginnings and ends of the read. So they're not the sequences of interest for us. They're just some technical addition that needs to be removed, right? So then we use the PCR clean. Some sequences are uh, replicated with much higher intensity than others, and we have some sequences that are abnormally replicated, right? So we want to adjust for this. We use PCR clean to do that. Then we use a HESAT. Here you have the top hat, but we use HESAT in the demo pipeline, and HESAT is actually used to map on genome. Further steps were map isoform construction. So those things that we are mapping on the genome, we have a genome model, right? It could be a human model. There are options on the platform. We'll repeat all those steps later in the program. So there is mouse model or human mouse model, right? So you map those reads that you have from the sample on those models, and you use HISAT for that. Now, those reads could be just parts of the final product of expression. So those parts could be merged, they could be spliced in different way, and we have different products, right? So genes are not final product, right? So when you transcribe a code from gene, that is not a final product, it's processed. So when you process it by using different DNA enzymes like ligase, for example, or restriction enzymes that can splice the genes, we get different isoforms. So we used cufflinks and cuff merge for that. Then we used bowtie to map on the transcriptome. So difference is when we were mapping on the genome, we were mapping on all the genes. Now we're mapping only on final transcripts, right? So we get the final expression table, which is created by using RSAM expression table algorithm. Now, this algorithm has statistical perspective of finding the most expected probabilities for all the reads, and we are creating an optimized model for gene expression. So I just did a brief recapitulation of all those steps. You have all those steps on the T-Bio platform. We ran that demo 2 or 3 times, so I won't be going back to that again. If you have any questions regarding this, again, you can ask any of us. You can send us the mail. We can run the pipeline together. And that was the recapitulation of this. Now, I will introduce you to a new data set. It comes from a new project. As you have seen, we use different data sets, right? First time we used that 1000 genes data set. It had just gene expressions for 11 samples, and those samples were related to the breast cancer. But we didn't have any other information aside from samples names. Then we used PAM50 dataset to the t-test. Right now, the PAM50 dataset had some additional labels. We saw that we have gene IDs, we had sample IDs, but we also had some categories like normalized, closely low, like basal, for example. So we had categories, and those categories were used to compare samples. So we included them in the categorization of the statistical analysis. And such a data set is called a labeled data set. So it's labeled in a statistical way. On the other hand, we used the PDX model last time. We had two types of labeling. We had labeling of type of breast cancer, which was like ER plus or triple negative, and then we had a labeling regarded to the type of the mouse that was receiving that tumor. So we had those ATH mouses and NOD mouses, right? So we had two layers of labeling, and we used them to conduct a factor analysis. As you can see, this is the project overview of our new data set. So it also has those SRRs, right? So SRRs were the source of all the information that we are extracting the data. Now, there are a lot of information here, but I want you to pay attention to last two columns, which say cell type and weeks, right? So all of these are certain labels. Cell type is dividing the data set in the cell type categories. So these labels tell us if a gene expression comes from MAC-A, MAC-B, or MAC-B2. MAC represents a macrophage. So we're not analyzing cancer expressions now. We're analyzing expressions from macrophages, which are immune cells, and we'll speak about them more during the session. Why are we using macrophages? Well, in our previous sessions, we were analyzing gene expressions of tumor cells, and that is completely logical. If you're analyzing the tumor, it's logical that you want to take those tumor cells, extract them from the biopsy, extract their gene expressions, and then put a table out of them and analyze. It's completely logical. But the tumor itself is not consisted just of tumor cells. A lot of other types of tissue and cells are included in the tumor. And these other types of cells are called the stroma or microenvironment of the tumor. So, a lot of types of immune cells are included in the tumor, which is completely logical because tumors are eliminated most of the time, or tumor cells are eliminated most of the time from the body by our immune cells. Sometimes that tumor is not eliminated, it overexpands, it proliferates, it creates its own microenvironment, and then it becomes dangerous, right? But our immune cells are generally fighting against the tumors all the time, and they are a defense line from tumors or cancer cells that arise in the body. So, what is the logical explanation? Why would we find immune cells in the tumor? Very simply because immune cells are meant to recognize the tumor and infiltrate it and destroy it. So in most of the tumors, we will find these types of cells. So there is another issue that tumor is creating its own microenvironment that is ideal or optimal for its growth. So it will produce growth factors that will grow blood vessels to the tumor. So it can have nutrients from the blood. It will produce many other molecules that will make this stroma of other cells its ideal environment regarding the nutrients, regarding the proliferation. It will make those other cells proliferate as well. So the tumor is growing from the tumor perspective, but also from these other cells that make the stroma of the tumor. We said that immune cells are infiltrating the tumor to destroy it, but some immune cells are just part of the stroma, so they're pro-tumor oriented, right? Some are fighting against the tumor and some are pro-tumor oriented. And today we will be using machine learning to analyze one type of cells from the tumor stroma, which are called macrophages. Macrophages are cells that destroy foreign bodies in our organism. They're compared to the other cells, they're quite big. They're 3 to 5 times bigger than most of other cells in our body, and they have a function of cleaning foreign bodies, destroying bacteria, for example, or recognizing and destroying tumors. So it's logical that we'll find them in the tumor. But how do we differentiate them? Are they fighting the tumor, or are they just a part of the tumor microenvironment that is proliferating along with it? Well, we will use those genes that we extracted from the sample and created a gene expression table out of them. And using machine learning algorithms, we'll create a network of interactions that will identify certain processes that are happening in this macrophage. So when we identify those processes, we would be able to tell if this macrophage is, for example, fighting the tumor. Does it have processes that are related to that, or is it just proliferating, feeding out of the nutrients from the microenvironment and playing along with the tumor? So, as I said, we have gene expressions from macrophages, and we have two labeling layers, what type of the macrophage, and we have those weeks. So some of them are measured initially when they're implanted in the tumor. Some of those gene expressions are measured 2 weeks after implantation, and some of them 3 weeks. We'll be using 0 weeks and 2 weeks in our data. So you receive the data set that has all these characteristics. It has names of those macrophages, names of the samples, cell types, and it has weeks. The only thing is that it is transposed, and I will explain later why did we transpose that. As we said, when finishing the NGS pipeline, when finishing the data science part, summarization part, we will come to a part where we want to identify certain factors that are influencing the gene expression of certain genes. So, in this case, one of our factors is weeks or time. So we can see, for example, that in these heat maps, you have heat map, we said that green is high expression and red is low expression, but in this case, it's turned around. It's just opposite. So we have a situation where weeks or time is influencing these 8 genes. You can see on the top left quadrant. They are increased in expression over time. So just after 2 weeks, 14 days, we have a high increase in expression of these genes. Some genes follow different patterns, right? So some genes are not connected to the time. They're connected, for example, to macrophage A or macrophage B2, right? So in this top right example, you have B2s that have low expression, and A subtypes of this immune cell has high expression. So we used factor analysis to identify genes that could be related to some functions of these macrophages, related to these factors as well, to the factors of time. Time is related to the tumor microenvironment, right? So if it's just implanted, it's not yet influenced by the tumor, but after 2 weeks or 3 weeks, tumor microenvironment will be a factor that is influencing gene expressions of these macrophages. And this time is a very important factor for us. So we won't be going through the factor analysis and regression and everything again. We finished this part for this data set, and now we will move on with the next part of the analysis that includes, as I said, machine learning, and it's called network analysis, or creation of Bayesian networks in this case. So what will be the product of finishing this analysis? We must have some goal, right? So we're not conducting this analysis, I don't know, just because of the analysis. We have some goal. Our goal is to create a network of pathways that will enable us to identify some processes in the macrophages. For example, is the macrophage proliferating? Is the macrophage, I don't know, is the macrophage using glucose metabolism, right? Is the macrophage uh, creating some immune response to the tumor? To identify these processes, we'll first need to create some interaction networks between those genes that are, for example, highly expressed. So, out of the whole data set that has, I don't know, 80,000 genes, we selected using factor analysis, BSEC, and other analysis to narrow down the analysis to those genes that are highly expressed, for example, that has normalized expression over 7, and we also added some interesting genes that showed up in the factor analysis. So in the end, we created a data set of about, I don't know, something less than 100 genes that you have in that data set that Pratim provided the link on the beginning of the session. If some of you didn't download the data set, you can do it now. You have the link on the beginning of the session, and if you can't, I don't know, if you want me to post it again, please just type in the chat or unmute yourself and I can post it again. So in the end, we'll be creating not networks like this. These are some huge networks. We'll be creating a bit simpler networks to be used for this educational purpose, but networks can be very, very complex. Now, I will just share my screen and go through the data set a little bit and show you how it looks like. So we said that we could use various software to view our data set, right? So we were using Excel, right? We were using RStudio to view the data sets, right? So, for example, I will show you just a second. Sorry. I will just reload this again. So I will show you, for example, how to use RStudio to view datasets. You have some datasets here. I will actually leave this one here, right? So we have the PDF dataset, right? So we have some other datasets. We had, for example, this is the dataset that we are using. So we said that we always want to use these options. Use first column and heading yes. When importing the dataset, you can see that this dataset is transposed. So sample names are here and gene names are here, right? So in our previous dataset, our sample names were here and gene names were below, right? So we just transpose that because algorithm that we are using requires us to transpose the data. It can be easily done in Excel. You just copy the data and you paste it as the transposed paste. So I won't be going through that right now. So we have various, these are not the samples, right? Some of them are samples, but some of them are same samples after 2 weeks, right? So we have 6 samples and those same 6 samples after 2 weeks. Now, statistically looking, we can view this as 12 different samples, right? Because gene expressions will vary, right? They won't be the same. They are related because they're from the same sample, but statistically speaking, they will be completely different. So, as I said, this is the labeling. We have MAC-A, MAC-A, right? And we have MAC-B2. So we have 2 types of macrophages, and that is 1 layer of labeling. We also have 0 weeks and 2 weeks, so that is another layer. Similar situation when we have ER plus, triple negative, and we have different environment of mouses in our last session. We had another type of data set. I'm just reminding you this. because we went through large number of data sets, we had this data set, right? 1000 cell lines. And that was our first data set when we conducted summary statistics. Take a look at it. It has gene IDs with ENSG, which said that when we have ENSG, that is a human gene, right? We had also uh, names of the samples, SRRs, and we had numerical values for them. But one thing is missing here compared to our previous session, for example, we don't have categories, right? So here we have categories. We have ER nod, right? So it's not just a sample name, it tells us that it's what exactly is that type of breast cancer. It's ER plus, or ATG and nod tell us that it's exactly that type of mouse model that tumor was implanted in. But if I take a look at this SRR, it tells nothing, right? It's just name of the sample that is created in databases like NCBI, for instance. So you can have very similar samples, but you need to take a look at what information do names provide. If they provide some significant information that could be used to divide dataset into categories, that dataset is called labeled dataset. If it's like this, you just have sample names, and those names don't represent anything except the name, that's unlabeled data. And there are two types of machine learning that could be used to analyze these two types of data. So I'll just go to the server. Again, Pratim, if you can provide them with the link if someone doesn't have a link to the server, but I assume you already have them all and you have used the platform various times. And here are two types of machine learning that could be used on T-Bioinfo platform. They're here under the data mining section. Supervised analysis would generally be used on data sets like this. We have that labeling that could divide our data set into categories, 0 week, 2 week, different types of macrophages, etc. But we could use unsupervised machine learning, for example, on a data set like this. It has no labeling, just sample names. There is another interesting situation. Unsupervised machine learning could be used on label data as well. So unsupervised machine learning is polyvalent, right? It's not just it's ideal for unlabeled data, but it can be also used on labeled data because you can expand something from that label, right? We have some groups here, but we could use unsupervised machine learning to find even more groups. But the situation with supervised analysis is different. You can't use supervised analysis on unlabeled data. So that is just some basic rule I want to teach you for now. And we will speak about this in our future sessions. For now, we will use a method that is universal. It's called, when we go here to unsupervised, it's generally used in unsupervised, but it could be used in supervised as well. It's called a Bayesian network. So as I said, it's a universal algorithm. It could be used on both labeled and unlabeled datasets. Before I run the pipeline on our dataset, I will show you something that could, for example, be, I don't know, an issue for some of you. You noticed on our last session that, I don't know, I ran few DSEC pipelines, but then when I was uploading the SVL file, something glitched, right? When I pressed, I don't know, file upload, let's use, for example, I don't know, this dataset that we are using today. It's called dataset macrophages. You'll see that some two files have loaded, and if I try, for example, to run, it can't, right? So it stopped. That's because you didn't wait for the interface info that was loading to finish, right? So if you do this immediately, sometimes you might come in a problem like this, where you didn't wait for that interface to finish what it was loading. But there's a very simple way. I assume a lot of you have figured out how to solve this. Way is very simple. So you just do this. You could potentially just reload the site here, right? So, and then you click start. So you load that interface and let it finish, and that's it. You solve the problem, right? It's very simple. Now you can upload the files, and here is the file. It will now create a pipeline, right? So you can see all these yellow buttons that are now available, and I can use this algorithm. So it's very simple. You just reload the site and click start, and it will finish everything for you. You just close the window. Some of you won't come into contact with this issue. Some of you will. If you, by chance, come across this, it's very simple. Just reload the page and click start. And then you can upload the file again. So now that we created, we pressed the button for Bayesian network, we have some brief explanation of what Bayesian network does. Imagine that these nodes are genes. They're this, right? So they're genes. Now we're not analyzing comparisons between samples as a whole, but we are narrowing down even more from differential analysis to these single genes and relations that they could have between each other. So we are not comparing genes across the samples. We are comparing relationships between the genes themselves. And we have different types of categories between those genes. We have their expressions at 0 weeks, we have their expressions of 2 weeks, and we are allowing those expressions to change over time just by having two types of variables, 0 weeks and 2 weeks. We are allowing them to change over time. So we're including all of that. When we allow them to change over time, we can see if they're correlated. For example, if something is increasing at first week and then we see another thing increasing at first week, and then same thing happens, I don't know, at 10th day, at 12th day, and then something decreases, and then the second thing decreases as well, that means that they're following patterns of each other and that they're correlated. We also said that regression could be used to analyze this correlation, and this analysis is closely related to regression. It is not regression, it is similar, right? There is big difference that when you run a regression, you need to normalize the data, right? So we saw that when we ran first regression last time, it was on normalized data 2 sessions ago, and we had that fine scatter plot. We could compare it to that diagonal line. But then last time we used it on raw non-normalized data, and we didn't see anything on the plot. There was just some couple of blue spots agglomerated on the lower left, right? So statistically, it is the same. If you run linear regression, you want your data to be normalized, and linear regression is very sensitive to having normalized data. That is not the case with Bayesian methods. Bayesian methods don't assume that everything is equal, everything is normalized, and you have that bell-shaped curve and everything, right? So they don't need such an assumption, and that is why they're called Bayesian, because they can analyze some different types of I don't know, parts of data. You don't have to take the data as a whole. You can take just parts of the data and then make conclusions on some other parts of the data. I won't go into this mathematical part too much. We will talk about this more, of course, during the program, but for now, let's consider it as a method that is more universal than linear regression. And another thing it does, imagine that you have large number of regressions, right? So regressions between all those genes, between this and this and this, and you combine them into a network. So you create a network of regressions. As I said, those are not real regressions, they're Bayesian-based methods, but they're similar. Imagine that you can analyze association between all those genes and create a network out of it. Another dimension that Bayesian network will add it will create a certain hierarchy of those connections, right? So it's not A, B, C, D, and they're connected. It will tell you if A is, for example, connected to B, and B is connected to C, but A is also connected to C, right? So it will create a hierarchy or some pathways of those connections in the network. If you analyze scientific literature about this statistical method, you will see that these, for example, I don't know, this that we refer as genes is called nodes. So these circles are nodes, and these connections, linkages between them, are referred as edges. So you also have a textual representation of this below this Bayesian network, and you can read it on the pipeline. You see that green lines or green edges represent something that would be similar to a positive correlation. So when one is increasing, the one that is connected in the green line is also increasing with, right? So that's the nature of association. On the other hand, if you have red lines, it's opposite. For example, if this one is increasing, that would probably mean that this one is decreasing because a red line is between them, right? So it's just opposite direction. Red doesn't mean it's not associated. Red could be even more associated than green, right? So green is just positive association, which means that they are pretty much in the same direction, and red is opposite direction. You also have some uh, lines that are interpreted, and those are some smaller associations, and we have threshold for these associations. Now there are some standardized thresholds that could be used for each different data set, and generally we will want to use this setting. For example, we will set this one to 0.5. Now we know that 0.5 is not statistically significant value, so this is a soft threshold. We can include some non-significant values, but it's important to those that are not significant, right? So sometimes something that is derived from the t-test is not significant, but it could be significant in some Bayesian network, right? So we will use this high threshold. It's 50%. It's 50-50. Call it use a p-value of 95% probability that we are right. There are various methods here. We will explore each of them, but for now, let's keep it on hill climbing, right? We don't need to log transform the data. As you can see, it's pretty much on this dataset macrophages. You can see that it's normalized, right? It's from zero, I don't know, from 0 to 11. So it's already normalized. We don't need to log transform. There is an option of creating a large number of replicates of this data set and then analyzing it further and create a more optimized bootstrap data set, but we won't do that for now. So we just use these settings and finish the pipeline. After clicking end, we will have an option to create the name of the pipeline. So let's call this one, for example, bias network, and it's for the macrophages dataset. So, sorry about this. So we finish the pipeline. We can see running here, and we just click Run Pipeline. So pipeline will start. As we talked last time, pipeline finishing time could vary from time to time. You can see that mine has started right now. This is the last one. It says started, right? So if you refresh this, you will see that it's finished in a couple of minutes. Now we'll get back here to see exactly that it's finished and how do we download output files. But before we come back here, let me just show you what is the output that is produced. oh, it's a PDF file. You will have some TXT files and you will have a PDF file that looks like this. Now it's zoomed out, right? So the PDF file doesn't tell you much information right now, but when you zoom in, you will see those networks. You can scroll left and right, and you can see that actually network is created from those approximately something less than 100 genes, uh, and they're pretty much all those important genes are here. We said that we extracted them by using some other analysis, but generally now we have a more depth, created some interaction networks between. Now, first we need to define this term interaction. When I say we have Bayesian network and it shows interactions between genes, it's a statistical interaction, right? So we use statistical models to create this interaction. This does not necessarily need to be a biological interaction. You saw that probably if you, I don't know, if you were in some biochemistry or molecular biology classes, you probably saw that there are various types of biological pathways, I don't know, signaling pathways for some cancer genes or some other disease type, or some processes in the cells. What is interesting here is that if we use a method like this, that is very accurate, as I said, uh, it's a machine learning model that extracts some relations in the data that we didn't have just by, I don't know, comparing expressions. So we say, hey, this expression is higher in one sample, it's lower in another, and we can't conclude anything else. But using a machine learning approach, we can create a network like this. And these networks are created from statistical models that are computed many times to create a most optimized model. So when you create that most optimized model, you create a very accurate assessment of interactions in a statistical way between those genes. The most important thing is that statistical interaction model will generally correspond to some biological process. And we will see that as we continue the session. So what I can conclude here is, for example, I don't know, this gene, some genes are from the mouse. You can see that in the data set we provided, most of these genes are with MUS, so it's spelled like ENSMUSG, and then we have some code, right? So when we have that MUS, it means that genes are from mouse. So mouse was the model from which these gene expressions are extracted. So these genes belong to macrophages from mouse tumor or macrophages from stroma or microenvironment from the mouse tumor. We could identify some processes here. For example, let's say that this is a certain process. We still don't know what is it, but we statistically confirm process is a part, I mean, all these genes are part of the same process, right? So they're connected. And when you have just, I don't know, two genes like this, it's pretty vague. It doesn't have to mean anything. Yeah, something is correlated to something. It can happen a lot of times. But when you have means that some process is identified. So we definitely identified some process that is happening in these macrophages. And if we found out what this process is, maybe we could identify the status of the macrophage. Is it fighting the tumor? Is it destroying it? Or is it just some part of tumor microenvironment? This is very important because when you analyze cells in the tumor, you want to know what are they doing, what are their functions, what is their status, right? Are they good or are they bad? Are they pro-tumor or are they fighting the tumor? Could you extract some type of cells from the tumor that could actually be used as the therapy? There are also therapy approaches that extract, for example, macrophages or lymphocytes from the tumor because we know that they're fighting the tumor, and then you multiply them and put them back in the body. So using processes like this, we can identify the status of that cell. How do we identify? Well, very simply. Let's, for example, take name of this gene, right? Now first let's see if the pipeline is finished. You can see it's done 100%. So now you have all those files and you have PDF file here. When you open it, it will download and you have the same thing that we were viewing right now, right? So how do I, as I said, identify what process this is? Well, simply, we have the process, we have the network that identified the process, we have a lot of networks as well, but let's say this one is most interesting, it's most complex. Well, first, for example, you can go to NCBI, right? And you have name of that gene. So I just got it here, and you can paste it in the NCBI, and you will find that gene. So you can see these blue marks here. So we found something. We found one gene, right? You can view that gene, and you can see where is it expressed, what it belongs to, just by going down here, and you have various information here, right? You can see expressions tissues here, right? You have some path right here, pathways and biosystems. Now, there are a lot of processes, as you can see, in which this gene is included. So we have here regulation of actin cytoskeleton, right? So we have some other things here, like interactions of photocytes, expanded and other, right? You have a lot of processes, but you also have some interactions here that could be further analyzed. We won't go there right now. We have other processes here, right? Like, I don't know, establishment of T cell polarity, right? You have platelet formation. You have phagocytosis. There are a lot of functions. To get to the real function, you must also analyze others and see what are they similar in. For example, right? We as something that could be interesting for us because macrophages generally kill cancer cells by phagocyting. We know that from the theory. So we identify this as something that could be interesting for us. We could do this in another way. For example, you could just type name of that gene uh, in Google and you will find a lot of these sites. And for example, you have this site, Ensemble, which is related to EMBL or uh, that type of institute that has databases similar to that to the NCBI, and you can view similar things here on Ensemble. You can also see uh, some things in the genome. We'll go through this later, so don't be confused by all of this. You have some legend here, and then you have some tabs here on the left. This tab is very important. It's called external references, right? So it tells you what references of bioprocesses are connected to that gene. And we have something here that is called the reactome. So what is the reactome? What are reactions or interactions between that gene and some processes that are happening in organ? So if we analyze this, we have here innate immune system, which is logical. Macrophage is a part of immune system, right? And right here, we also have some other processes, but we also have phagocytosis, right? So we found it 2 times. And we know that phagocytosis is something that macrophage could use to kill cancer cells. So we definitely identified a gene that is connected to regulation or initialization of phagocytosis, because here we see more information. For example, phagocytic cup formation. So it's like an initial phase. We had a lot of detailed information on NCBI. We have detailed information here. And you could combine these databases to get some assumptions. Now, we identified that it is connected to phagocytosis. So if I performed similar analysis of finding these annotations, analyzing references and analyzing reactomes similar to what I did now on Ensemble, I could create approximation of most relevant processes that are connected to these genes. So I already did that. We could go through each one of those genes, but that would be very time consuming. And I have created such a slide for you. So if you take a look at this slide, here is the first one we analyzed. We said that it could be connected to phagocytosis initiation. We could have a case where we use some of those other processes, and then we would find some illogical networks. But I took phagocytosis and I took other genes and extracted most relevant information from all of them. So take a look at this. Phagocytosis, neutrophil degranulation, energy metabolism, right? So it's logical. Neutrophils are also immune cells. Their degranulation is part of phagocytosis process. So they're logical, right? And energy is needed to do that. So cells should be saving the energy from other processes to be able to conduct phagocytosis. And these are some logical steps. So we initiate the phagocytosis, neutrophil degranulation is expanding on this process, and energy is being lowered in other processes of the cell to save it for this process. You can see that there is a red line here, which means that as this is happening, energy metabolism is going down. Take a look at these other branches. Interleukin signaling. Interleukin is signaling molecule that macrophages are creating to attract, for example, other cells to the tumor. We have neutrophils here, so we know that there are other types of immune cells that are infiltrating the tumor, and we know that there is some signaling that is included. Macrophage expressed perforance. That I found a lot of other things. You could find a lot of other things that are connected to immune system. Some of them are not even connected to the immune system, but it's logical to create a pathway from those that are related to each other. So we have macrophage expressed perforance. Perforance are used by T cells, which is a type of lymphocyte, to destroy tumor as well. So you can see that all of these functions are connected to destroying the tumor. We have phagocytosis, which is a process in which macrophage uptakes the whole cell of the tumor and then destroys it. You have interleukin signaling that is drawing neutrophils and, for example, T cells, and then they're also improving this process of phagocytosis and destruction of cancer cells. A lot of energy is needed for this, so we're saving energy. There is another interleukin signaling pathway. So we have two signaling pathways that originate from the macrophages, and they're also connected to other immune cells which are fighting the cancer. So here's another neutrophil degranulation. It was here, right? So we already have a lot of similarities here. All of these processes are either identical or they're causing each other in a biological way, and They're, as we can see, created by using bias network or a statistical network. So in this case, statistical model is very similar to a biological pathway. In essence, we use the statistical model, we use the machine learning algorithm called bias network to create a statistical pathway for these biological processes, and we can see that those two match. We have another here, which is called platelet activation, signaling, and aggregation. Platelets have various functions. They contain large number of growth factors, and they're also used to, I don't know, agglutinate tissue when it's under stress. We know that when we have some small wound or something, when it's regenerating, we need platelets to initialize that regeneration. And it's logical that if all this is happening, we have neutrophil degranulation, phagocytosis, a lot of stress in the tissue, a lot of energy is consumed, and it's logical that something is going to be needed to fix all these damages that could arise from neutrophils and macrophages destroying the tumor, or just some cells in the tumor. It doesn't mean that they're going to destroy the tumor, they're just activating this process of fighting the tumor. So I will just go through these things from some publications and then get back to it again. We see here phagocytosis of cancer cells. So you see macrophages and you see the tumor cells. Macrophages are uptaking the tumor cell in its own volume, and then they're destroying it. You also see some T cells here, they're co-stimulatory. We said that some of those interleukins that are connected to those genes are actually drawing these T cells to approach the tumor site. So, phagocytosis is a process of destroying the tumor cell with many processes. We have tumor cell cytotoxicity. We have the phagocytosis process itself, but it's facilitated by some other cells, like T cells or neutrophils. Here are neutrophils. We found that two of those genes are connected to neutrophil degranulation. What neutrophil degranulation does, it creates this phagocytic cytoplasm. What that does, it creates traps for tumors. When they come across them, that will be a signal for macrophage to phagocytize that cell, to destroy it. So you can see that from biological and biomedical research, there is a whole foundation for this pathway that we identified, and that all of those processes are interconnected. So we have phagocytosis initiation. We saw that this one is connected to that first stage of forming the phagocytosis cap. So it's also first in our hierarchy. And then we have some branching here. We have two types of signaling. We have neutrophil degranulation, some other molecules like perforins connected to cytotoxic cells, some platelets, and saving energy for the whole process. We saw that all of them are closely related as a biological path. So we could expand this analysis on other parts of our data. We have that this is the part of the network we were analyzing, but you could analyze this one, you could analyze this one, you could analyze any of them. As I said, those that contain just two genes are not very relevant, right? So you might come with some false results here. But if you have a complex network like this, or like this, especially as the one we analyzed, which is including 9 genes, that means that you definitely have some process. To just recapitulate on this is a Bayesian model. It's very accurate. It doesn't have many assumptions of normality of the data, of some other characteristics of the data we need to check. It's Bayesian. It could be used on pretty much any data. And that is why it's universal. It could be used on both labeled and unlabeled data, and that's why it's like the first best algorithm for us to use, because there is a small probability of making a mistake when analyzing types of data. Now, network is created by analyzing associations and statistical interactions between the genes themselves, not between different genes in different samples, but between genes in the same samples or same genes in the same sample, for example, in a different time frame, right? So we created a network with these nodes that contain name of the gene, and we have some green or red lines with arrows. Green means that they're associated in a similar positive way. So when one is higher, the other one is higher, right? And red means that they're negatively associated. They're opposite. When something is increasing, some process is increasing, the other one is associated with it, but it's decreasing, right? So right now, we will stop here. So we use first machine learning algorithm. We will start with some detailed explanation of each machine learning algorithm, and we will start with the PCA next time. We will also create some videos for you that will explain some statistical perspectives of what machine learning is and what is the main difference between machine learning and mainstream statistics. But for now, we will stop here. We have shown you how machine learning can be very effective in identifying some biological process. So, in essence, we identified a biological process. It's called phagocytosis and some other processes that are related to it. And this means that this macrophage is actually fighting the tumor. It's destroying cancer cells. It's useful or it's positive if we analyze its properties related to the condition or the tumor. So we now answered that main question. We took a macrophage from the tumor microenvironment and we were interested, is that macrophage just proliferating with the tumor? Has it become a part of the microenvironment? Is it negative for the host? Is it pro-tumor active? Or on the other hand, is it actually fighting the tumor? Has it recognized it? And is it drawing other immune cells to destroy the tumor too? So the answer is very clear. Phagocytosis, interleukin signalings, neutrophils, drawing other cells. Answer is clear. This cell is fighting the tumor. And for example, such cells are very interesting in biomedical research, because if we know that cell has recognized the tumor, it's destroying it, perhaps it could be multiplied, it could be augmented to fight it even more, it could be analyzed to see how has it recognized the tumor, many other things, and expand the research from this perspective. So right now, before we go to something that we'll do in our next session, I would like to leave the stage to you. You can ask any question that is related to anything, to previous sessions, to this session, to anything that you don't understand, to the pipelines, to the theory of all this. And please unmute your microphone or just ask a question in the chat.
Darko, there's a question in the chat. Can you address that?
Okay, thank you, Pratim. So we have a question from Ambi, right? So for constructing a pathway through supervised or unsupervised analysis, is it always necessary to work with gene expression data? What other types of data can be used to construct pathways, or just relation to the dataset? This is a very good question. So far, we used gene expression data. We said that we have gene IDs, we have samples, we have some labels, some categories, right? Some time frames, 0 weeks, 2 weeks, and then we have numerical data for all this, right? We could use factor analysis to analyze this. We could use other types of analysis, but we're dealing with gene expression data. Why did we use gene expression data so far, and why is it so successful, for example, in this case, to define a biological process? Well, because gene expression tells us a lot of things. First, it tells us that some gene is connected to something. Some gene is different than some other genes, or it's connected to some genes. On the other hand, we have the exact expression level of each gene. So we have huge amounts of information. We saw that this information needs to be processed, etc., etc., and then we could use machine learning. So transcriptomics types of data that contain gene expressions, is very useful because it's very important. But there are other types of data that we could use. I will show you, for example, here on ENSNBL, we have, let's, for example, select a gene. I don't know, let's select one of those genes. Let's select, I don't know, this one. So we can review that gene not just from transcription perspective, but we can view a lot of other things, right? So, for example, we could view some isoforms. We could view coding parts and non-coding parts. We could use a lot of these things. We could, I don't know, expand on this analysis. We could see exactly where it's located. We have many other things here, right? So we could, for example, analyze single nucleotide polymorphisms, right? So each one of these polymorphisms tell us, for example, that some gene could have a single change, A or adenine is changed to thymine, right? So it's changed exactly in some location, and that location could be, for example, one of our genes. It's logical that these genes are related, for example, to fighting the cancer. We could analyze, for example, SMPs in this gene. We would just, for example, find this gene and see what SMPs are present. Could some of these SMPs be added to the whole data set so that we don't have just expression levels? Along with this, we could have SMPs. And we could visualize all of that on the genome. We have percentage of GC pairs, for example, in this gene. We could have a section of GC pairs here, right? So there are many other things. We'll talk about all of them during the program, especially when we start accessing the data. Some of you might choose different types of data. You might choose SMP data, for example, or single nucleotide polymorphisms, those small changes in the gene, those small changes that could change the function of the gene. So, for example, imagine that some change has happened here and this gene is not working properly. That probably means that immune system would be worse and this whole process wouldn't be functioning very well. So a lot of information could be added to just this transcriptomic data, and we'll speak about them during the program. In this case, I've shown you SMP. How can we identify those SMPs? We could, for example, I don't know, we could identify many other things here. We could zoom in on this, for example, and we would probably have a better perspective on those SMPs. Take a look at them. Now you can see each SMP individually. Let's say, I don't know, this change is interesting for me, right? So maybe I could analyze in those 0 weeks, 2 weeks, M1, M2, B1, B2, and add this dimension. Do any of these samples have some polymorphisms? Is some gene connected to something more if it has some SMP or some change in its sequence. If, for example, A is changed to T, perhaps this gene could be related to something else, right? So we could expand on this. There is also a way to do this on the T-binding platform, and we'll also talk about this during the course. We will have exemplar projects, and if we go to areas of analysis, you can do this on your own as well for now. There is something here that is called a mutation variant. We all know that mutation is something similar to this SMP. It's just change in, I don't know, larger or smaller number of nucleotides, and it's changed because of some radiation or some environmental factor or something else, and it's called a mutation. So we can analyze mutations as well. Here we have a demo on variant calling for those mutations. You have a lot of these algorithms, and for example, if you have explanation here of what are codons and amino acids. Three nucleotides code one amino acid, and each of those nucleotides, if changed, could create a different codon, and it could produce a different amino acid. Now, different amino acid in the protein could mean different function. It could change the function of proteins that are created from these genes. So you have explanation of all of this. You have all those steps. You have variant calling, pre-processing, which is similar to what we did in NGS. We also have this similar part. We have the same save algorithm pipeline. Its pipeline is a bit different, but we have the same algorithm about I2, which is used to map on the transcriptome. And then you have some additional algorithms, variant calling. You have annotations that could also provide you with information about the genes. For example, I don't know, if I start this pipeline, if I started this pipeline, just a second, you click OK, you have option to do two. Let's, for example, just go directly to about I2, right? So this file is already PCR clean, so we don't need to do that. We just go to about I2. And this also explains to us what about I2 does, how it maps those reads on the transcriptome, and then we can go to visualization. So we got the similar picture to the one that we got before. You also have here some genes, some reads, and then you have those brackets, those lines or small tabs that represent SMPs or mutations. In this case, they're mutations. We run mutation variant calling algorithm. So these are the algorithms that could be run to identify these mutations. We didn't run it yet, so we just visualized potentially the sites that could be identified as source of some important mutations. Now, they could be analyzed further using a lot of these algorithms, and when we run this pipeline, for example, we will get a similar situation to the one that we got here, right? So we would view the picture that is similar to this one. We saw these brackets that are, for example, on this ensemble site. They also appear right here. I don't know if you can see that. If you can see that on, wait, I'll just run this again so for you to see, and you can see them here as these small lines, right? So you also have some, I don't know, scale here. We'll talk about that later. We'll have projects. We have that microbiology project that involves variant calling, and we'll speak about that later in detail. For now, we will stop here. So aside from, for example, SMP data or mutation data or I don't know, transcription data that we dealt with mostly so far, we also have the FASTA files, right? So FASTA files are sequences. We said that those FASTA files are actually codes of A, T, C, and G, which are part of the DNA structure, but they could also include U letter, which stands for uracil, and it's included in the RNA. So we have sequence data as well. Sequence data, sequences could be also added right here. We could, for example, I don't know, we could take this gene, right? Let's take it, and then go to the NCBI. So if we go to the NCBI, we could, for example, I don't know, we could find that gene, and we'll be able to find, for example, I don't know, we have a gene, and we would be able to find a FASTA sequence for that gene. We just click FASTA here, and it will give us the exact sequence for that gene. So now we have multiple dimensions of analyzing genes. We have the whole code, right? We could combine this analysis of this code, which will be used using BLAST algorithms, which could be also run on NCBI. They could also be run right here on the TBioInfo platform. So we won't go further into this. As I said, multiple types of data. There is even the visual type of data, and we could combine them. We'll speak about each of those types of data later during the program. So now, please, if you have any more questions, if this is unclear to you, you can ask.
Yeah, Darko, quick questions from me. This is Pratim.
Okay.
So the network that you showed us in the PDF file.
Okay.
Now there were some. Okay, so the arrows. Does it mean that the gene which is on the top, this is modulating all the other genes at the same time? And this is kind of fighting the cancer, which means that they are trying to modulate the expressions of the next gene. So so this network also tells me that there can be the this can be a basic process to identify the potential biomarkers for if this process is fighting the cancer, then I can help the fighting by designing some therapeutics. Am I correct in that way?
Yeah, you're absolutely correct, and that is a very good assumption. So in this case, we can see that this gene on the top is related to all of these three genes. Now, that tells us that there is high probability that it is the central point, right? It is part of the process that is modulating all of these processes. Now, it doesn't have to be 100% accurate because we are still dealing with the statistical model.
Correct.
As we saw, a lot of these genes have other functions, right? So it needs to be confirmed biologically and combined with this statistical model.
Exactly.
What this statistical model does for us, it identifies, for example, 9 genes that could be associated. There is very high probability that these 9 genes are part of the same process, and this gene on top is actually in the initial phase of the process. If we had more genes, maybe we have some genes that we didn't include in the dataset, right? Maybe
Correct.
there is another dataset, and maybe there is another type of tumor. So a lot of these data could be combined to confirm if, for example, this gene is the driver of the whole process. There could be some other genes that could be above this one. But here we have a clear picture that we identified 9 genes that are part of the same process, and we can say with the highest probability that this gene is something that is in the initial phase of phagocytosis. We have that reference that is actually included in the first stage of cap formation when phagocytosis is initialized. And then we have these genes that are related to actually some other cells, but they're all related to this gene. So phagocytosis initialization is also followed by some other processes which we saw in that file right here.
Yes. Yeah,
that's a very
absolutely.
good question.
One extension. Absolutely agree with you,
Makes sense,
Darko.
right?
So one extension to the question itself is that, you know, since these genes are correlated, so we find that these genes seem to be correlated with each other in terms of function. Can we perform a correlation coefficient study on this, like, you know, deriving the Pearson correlation coefficient or
Yeah,
the other one?
absolutely. And this is the result that you would get.
Okay.
Uh, since I said that this is not the actual correlation, this is something that is similar to the correlation because it's Bayesian method. And correlation, for example, Pearson correlation is a linear method, right? This is not exactly 100% linear since it's Bayesian, but they're very similar. If you run a correlation, for example, I'm not going to do that right now, but if you did it, if you, for example, took, I don't know, identified that gene, I don't know what is the name, we would need to find it here. And if we found it, and if we run correlation between two genes, let's say that those two genes are these two,
Correct.
right?
Yeah, yeah.
If we run the correlation, we would find the correlation like, I don't know, 0.7 or
Yep.
0.8. It would be a very high correlation. uh, we know that correlation is from 0 to 1. So 0.8 is high correlation. But there is another interesting thing. If we run a correlation on these two genes, let's say that these two genes are somewhere here. I'm not going to search for them right now.
Sure.
Let's say that these are two genes, these two, right?
Hmm.
So if we run a correlation between them, that correlation would also be high, but it would be negative. It would be
Exactly.
minus, it would
Yeah.
be minus 0. I don't know, 6 or 0.7.
Yeah.
So that is definitely the case. We would find correlations between each of those genes. But if I run a correlation between this one and this one, I would probably find no correlation or no
Right.
significant correlation. So
Exactly.
this network is very similar, and it's a very good thing that you mentioned that, Pratim. You could consider these as some types of correlation. But please keep in mind that these are not the actual correlations
Correct.
and that they are derived from the Bayesian statistical methods, and we will speak about those methods later during the program.
Perfect.
Makes sense,
Uh,
Pratim?
Absolutely. And one important thing you mentioned about it, that when the number of nodes are two and there's only one single edge, is there anything which is called minimal size network?
Well, in theory, there we can't consider there are some thresholds. We saw that there are those
Yep.
p-value thresholds,
Yeah.
right?
Yeah. Yeah.
There are, in theory, there are different methods that have, for example, I don't know, it's best to use if you have 5 nodes that are intercorrect.
Correct.
But there is no standard thing like that.
Right.
Sometimes you can use just 4 nodes. Sometimes you want to use 55 nodes, right?
Yeah.
But if you have 2 nodes from the experience perspective, I can tell you that it shouldn't be considered as something important. Here we have
Yeah.
just 2 nodes.
Mhm.
Why is this? Let's go back to the correlation again. Now, some things could be correlated not because they're directly associated, but because they're associated with some third process, right? So, for example, I don't know, you have one process and it's correlated to height. I don't know. Let's say you have height and weight, right? And they would probably be correlated, which doesn't mean that height is the causal factor of weight, right? They're just connected to development of an organism, and as we grow, our height and our weight are both increasing, right? It doesn't mean that they're causing each other. So they could cause each other uh, in some aspects. For example, it's logical that if you have higher height, you will also have higher weight. But it doesn't have to be the case. You could have a situation where you don't have to be very tall and you could still have increased weight, right? So they're not causative to each other, but they would definitely show correlation, and they're both connected to the same process which is called development of an organism. This is the same situation here. We could have different correlations and they don't have to be connected to each other exactly, right? They could be connected to something absolutely unimportant for us, and that's why they're showing some level of correlation. But if you have 9 genes and all of them are intercorrelated with each other and they're actually all correlated to this one gene, so these 8 genes will show some level of correlation with this one, right? Now that's a very strong network, and it has very little probability that this will be just some, I don't know, random correlation because some other. So that's another very good question for team, and it's very important to for students to understand this. If you have a complex network, that lowers the probability of finding false correlations. That is why we use this example to actually identify that process of phagocytosis and not some of these.
Thank you very
Okay.
much, Darko. It was a wonderful, yeah, thank you very much. Thank you very much.
Thank you so much, Pratim. Let's see
Yeah.
question from Sunita. So all our study pertains after splicing the RNA format to mature RNA, we have isoforms. Not all, right? So as I said, we will have projects that won't deal with RNA at all, right? We will have VCF files, for example. I showed you those mutations, variant callings, and we will have that example in one of our exemplar projects about Ebola and microbiology. But we could use we will use FASTA files. FASTA files are just sequence files. They're not transcriptome related, right? So there are also other types of visual data, numerical data. There are also one of the most important types of data. And thank you for that question, Sunita. One of the most important types of data is labeling data. As I said, a lot of information can be drawn from these categories. So we have 0 weeks, 2 weeks. We have MAC B2 or MAC A, right? So a lot of information could be derived from that. Or here we have ER nods and then we have ER ATG. We have different categories. That's also information, right? There is another type of data that we'll also be dealing with, and it's called annotation. So these annotations are actually showing us what are these genes related to, based from the scientific research, publications, bioprojects, and that. So there is also annotation algorithm here on the KeyBind platform. It could be also found on these databases sites like NCBI or EMBL, and we will also talk about those types of data. So there are multiple types of data. You have FASTAs, FASTQs, I don't know, transcriptome type of data. You have sequences, genomic type of data. You have methylation type of data, right? So you have some changes to that DNA sequence. Then you have visual data, you have annotation data, you have categorization or labeling data. You are combining all of them to find out something about the gene or disease or process that you're analyzing. To do all this, you need very complex algorithms, which are called machine learning algorithms, like, for example, this one, PCA, or the Bayesian network we created. And that is the point of using these algorithms. You have various types of data and you're integrating them. You can't integrate them using a t-test, right? You can't integrate them running a simple correlation. You need machine learning algorithms to create, I don't know, these networks out of more types of data. Now we use just transcriptome type of data, but we could use mutations, we could use sequences and add them to these transcriptions or gene expressions, and then create a Bayesian model out of all those types, right? So we're not limited to just using gene expressions, but as I said, gene expressions are very informative. They tell us what gene is expressed in what tissue and exactly in what amount. So that is one of the most informative types of data in molecular biology. But as I said, there are many other types. Thank you, Andy. So as Pratim mentioned, there are also protein sequences, right? So we can include proteomics, right? So we don't need to have just DNA or RNA sequences. There are also protein sequences or change in the protein sequence that could cause, I don't know, some disease to be more aggressive or to change in some other manner. So all of them can be analyzed using machine learning algorithms, and that is the whole point of doing these uh, precision-based uh, biomedical statistics, right? So we need to have large amount of data, and we have them, right? So there are large there is a large number of types of data on all of those repositories, and a lot of it is available from, I don't know, more than 100,000 bio projects on NCBI. So a lot of that data can be integrated, analyzed, and compared to some clinical data or annotation data, right? It can be connected to identify biological processes or find subtypes of breast cancer. We had, for example, those, I don't know, luminal and normal-like, but we could find even more using unsupervised analysis. And we'll show you how to do that in our next session, which will be about dimensionality reduction. Any questions before I go to this last part? Are there any more questions related to everything? Okay, thank you. Thank you, Sunita. Okay, please, if you have any questions, unmute your microphone or ask a question in a chat. Thank you. Thank you so much, Pratim. Thank you, everyone from the team. Okay, so next session will be held on October the 7th, and we will use another type of machine learning. This time, we will be differentiating different machine learning algorithms and how do they work. So sometimes we have very complex data sets. We have huge amounts of genes, samples, or subtypes of the disease, or other types of data, and we want to simplify that complexion, right? We want to select parts or portions of the data that are most important for us. But we now want to use machine learning to do that more accurately. And we will talk about that in our next session. If you don't have any more questions, I would like to thank everyone. Thank you, Sarah. Thank you, everyone. Thank you, Andy.